Hello everyone, welcome to my YouTube channel. Today we will be going over my notes for subtopic 6.1 of the IB biology syllabus. This subtopic is about digestion. So the first thing we would need to know about digestion is what it is and what is it for. Digestion is the breakdown of big molecules into smaller molecules that you can absorb. For example, if you ate an apple, you wouldn't be able to let it go directly to your bloodstream. You need to digest it first. You need to break it down into smaller nutrients that you can absorb. There are many steps to digestion. However, the ones that we will cover in these notes and in the IB syllabus are digestion, both mechanical and chemical, and absorption. Okay, let's get started. The digestive system is basically a tube through which food passes from the mouth to the anus, so that's what we're going to cover. The first part through which food enters is the mouth. You know that in the mouth, mechanical digestion happens, also known as chewing, and the food mixes with the saliva, which comes from salivary glands. This creates a food mixture called the bolus. This saliva also has some enzymes which start to digest starch. I will cover that later when we talk about the different enzymes. But yeah, basically salivary glands start the digestion of only starch and creates the bolus. This bolus enters the esophagus, which is also known as the gullet, and it is moved down by peristalsis, which is the movement of food throughout the digestive system thanks to muscles. I will also explain what peristalsis is in more depth later. But the epiglottis is a small flap because in your throat you have two channels, one for breathing and one for swallowing. So when you swallow food, the epiglottis closes over the windpipe to prevent that you choke because the food enters your airways. So after that, after the epiglottis closes, so food can go down your esophagus, there are a series of muscular contractions, which is called the peristalsis, which delivers the food down to your stomach. So that was the first transport of the food, and you probably know, if you have been studying the IB biology syllabus and you're revising now, the pH of the stomach is very acidic because there are many enzymes and acids there that kill the bacteria and other pathogens that might be found in food. And also these are the initial stages of protein digestion because they become denatured. Then the food passes on to the small intestine. Of course, now the liver and the pancreas intervene, but let's just imagine the track that I will show you later. The food moves to the small intestine, and these are the final stages of the digestion of lipids, carbohydrates, proteins, and nucleic acids, with the enzymes that I will talk about later. And the nutrients are also mostly absorbed in the small intestine. The pancreas and the salivary glands and the bile duct and the liver and the gallbladder, all of those are secreting organs. They secrete or store enzymes that will be used for digestion. And the large intestine reabsorb the water. Pancreas secretes digestive enzymes such as lipase, amylase, and protease that I will explain later. And the salivary glands, as I explained before, they start digesting starch, and it also creates the bolus, but by moistening the mixture, so it's not so difficult to swallow it. The bile duct transports bile, which is another juice full of enzymes which is used for digestion. It is synthesized in the liver and it is stored in the gallbladder. Okay, so this is basically what it looks like. Okay, so this is what the digestive system looks like. We can see the salivary glands near the mouth and the tract of the esophagus, how it goes down to the stomach, and from the stomach it passes on to the small intestine, from the small intestine to the large intestine, and then to the rectum. But also in the middle we have the liver, the gallbladder, and the pancreas, which secrete the enzymes into the lumen of the small intestine. The lumen is the inside part of the intestine. So this is a microsection. I believe it is also taken from Bio Ninja. Yes, I forgot to mention it. This This diagram and the next diagram are both produced by Pianenja, just for the record. The structure of the wall of the small intestine, it's specialized to be more effective in the absorption of nutrients because it increases its surface area. It has mainly 4 layers, the villi, the mucosa, the muscularis, and the serosa, but the mucosa is divided in mucosa and submucosa. The outer part is the serosa. It is a wall of cells which is used as protection, and it is made of connective tissue. The muscularis, as the name indicates, is a wall of muscle cells. We have 2 types of muscle tissue within the muscularis, the longitudinal muscle, which you can see next to the serosa, which is used for peristalsis, and the circular muscle, which is used to contract so the food cannot be pushed back towards the mouth. After the muscularis, we have the submucosa and the mucosa. The function of the submucosa is to divide the mucosa from the muscularis. It doesn't do much else, and the mucosa is also connective tissue which absorbs the nutrients that are absorbed by the villi. The villi are small finger-like projections. They are very, very tiny, and they are used to increase the surface area of the intestine. As you can see, there is much more surface area with the villi than without the villi. That is how food is absorbed in the small intestine. However, how does it get there and how does it move? It moves through peristalsis, which is defined as the movement of food along the digestive tract via the contraction of sequential segments of longitudinal smooth muscle. So remember, the circular muscle does not do the peristalsis. As I explained before, the circular muscles contract behind the food, right? The food is going forwards, and in the back part of the food, the circular muscles contract in order to prevent this food from going back towards the mouth. These contractions are controlled unconsciously by the enteric nervous system, which is the most autonomous part of your nervous system because you do this automatically, you don't think about peristalsis. And during vomiting, abdominal muscles are used, so that's why food is pushed back. Peristalsis happens mostly in the esophagus, but it also happens in the intestine. However, in the intestine it happens much slower because you need time to process the enzymes in order to let them break down the food. Okay, next we have the pancreas. The pancreas has a small group of cells, which are called alpha and beta cells, which secrete insulin and glucagon into the blood in order to control the glucose blood concentration, but that will be talked about in topic 6.6, which is the hormone and hormone management. Now we will focus on the rest of cells, which are the ones which secrete digestive enzymes into the lumen of the small intestine. This process is also mediated by the enteric nervous system and some other hormones. These digestive enzymes are synthesized in the ribosomes and the rough endoplasmic reticulum of the pancreatic cells, and these are secreted by exocytosis. So when they show you a micrograph of a cell with lots of rough endoplasmic reticulum, as mentioned in topic 1, you should be able to identify them as a pancreatic cell because they need all that rough endoplasmic reticulum for the production of digestive enzymes and exocytosis. The pancreas secretes all of these enzymes into the lumen of the small intestine through the pancreatic duct, which secretes about a whole liter of pancreatic juice per day. And this pancreatic juice contains amylase, lipases, and proteases, which will be covered next. Okay, so the small intestine, we have many enzymes, and we have 2 types of enzymes, the ones secreted by the pancreas and the ones that come from this lumen. Okay, from the pancreas, these are enzymes which catalyze hydrolysis reactions, which is basically a breakdown reaction. Starch breaks down into maltose through the enzyme amylase. Triglycerides break down into fatty acids and a monoglyceride through lipase. Phospholipids, which are basically like the triglycerides, which are lipids but with a phosphate group, break down into the same thing, fatty acids, a glycerol, and a phosphate group through phospholipase. And lastly, proteins or other polypeptides are broken down into shorter peptides through protease. However, these pancreatic juice is not enough to complete the process of digestion. So there are many more enzymes that I will cover, because you kind of need to know them. Maybe they will ask them on a paper 1, or they will ask you to exemplify. Basically, you should know them. DNA and RNA are broken down into free nucleotides through nucleases. Maltose into glucose through maltase. That's all of this is topic 2 also. Lactose into glucose and galactose through lactase. Sucrose into glucose and fructose through sucrase. And the peptides that were broken down from the bigger proteins with protease are broken down into dipeptides through exopeptidases. And these dipeptides are broken down into amino acids with dipeptidases. You can see kind of a pattern in the enzymes, that the name of the enzyme is the name of the compound but changing an O for an A. For example, lactose and lactase, sucrose and sucrase. So if you forget it while you're in the exam, maybe try to remember the rule, and you will guess that maltose is broken down into glucose through maltase. However, there are some substances such as cellulose which can also be consumed. However, it can't be digested, and it passes on to the large intestine as one of the main fiber components in a diet. Okay, so we have covered digestion. Now it's time to learn how absorption works. Absorption is taking substances into the cells and the bloodstream in the small intestine, because it principally happens there, and the small intestine is around 7 meters long and 2.5 to 3 centimeters wide. And as I mentioned before, in this image of a cross-section of the small intestine, the villi are small finger-like projections of mucosa on the inside of this wall. They are used to increase the surface area. That's the main purpose, and they do it greatly. They increase it by around a factor of 10. They absorb products of digestion such as glucose, fructose, amino acids, fatty acids, and the free nucleotides that we mentioned before, but also they ingest mineral ions and vitamins. However, some unwanted substances are detoxified by the liver, and they do not go directly into your tissues, and the bacteria can be removed from the blood through phagocytic cells that are also in the liver. There are several methods of absorption that are the ones that you should also know from topic 2, but basically the nutrients must pass from the lumen of the small intestine into the capillaries in villi. The capillaries are these very tiny small veins that go all around your body. That is how nutrients enter the bloodstream. So firstly, the nutrients are absorbed through the plasma membrane of the villi, and then these nutrients pass out through allectial and the blood capillaries of the vilus. The 4 mechanisms that we will cover are simple diffusion, facilitated diffusion, active transport, and exocytosis. So we know some examples of absorption. For example, triglycerides. They must be digested before they're absorbed. Of course, because the triglycerides could be too big of a molecule to pass through a plasma membrane. So the fatty acids and monoglycerides are the product of the digestion. The monoglycerides can be absorbed through simple diffusion. However, the fatty acids need facilitated diffusion by fatty acid transporters. And once they enter the plasma membrane, they combine again to form the same triglyceride, because through this method they cannot diffuse back, because if not, the monoglycerides could just go in and out, and the triglycerides coalesce, so mix with cholesterol to form tiny droplets which become coated in phospholipids and proteins to form the famous lipoprotein particles. And this is the way many lipids are transported in our organism. These are released by exocytosis. So we have covered simple diffusion, facilitated diffusion, and exocytosis. And this is how they enter the blood and the lymph. Glucose, however, is polar and hydrophilic, so it cannot simply pass through the plasma membrane. Glucose is a little bit more complicated. Ultimately, glucose moves through facilitated diffusion. However, this facilitated diffusion is only possible if active transport happens first. So, we can say that glucose uses both active transport and facilitated diffusion. Because the sodium potassium pumps are in the plasma membrane, and these pump sodium ions from the cytoplasm to the interstitial places inside the phylum, and the potassium ions move in the opposite way. So they create a gradient in which there are more sodium ions in one side of the cell than in the other side. So the glucose can associate with a sodium molecule, and then as there is a gradient of sodium, they can move through passive facilitated diffusion. However, there was a sodium pump before, so that is active transport. And lastly, we will talk about starch digestion, which without the catalysts happens at very slow rates. And starch molecules are too big, so they have to be digested in the small intestine for them to be able to be absorbed. It's important to know that all these reactions are exothermic because they involve a breakdown of the particles. We have two different molecules of starch, the amylose and the amylopectin. Starch is the union of many glucose molecules, so that is the main difference between amylose and amylopectin. In amylose, the carbon 1 and the carbon 4 of the different glucose molecules are together, and using amylase, they can be broken either into a maltose and a maltriose, which is a maltose but with 3 glucose fragments instead of 2, and then maltose is broken down into glucose, and maltotriose is also broken down into glucose. However, amylopectin is the carbon 1 united with the carbon 6, and it is broken down using dextrins. This is broken down into glucose. So amylase can break any 1 and 4 bond, the carbon bonds, if there is a chain of at least 4 glucose monomers. So if you have 1, 2, 3 amylose chain, amylase cannot break those because that would be a maltotriose. However, if you have 4, the amylase can already break it down into 2 maltose molecules. And this yields glucose, which moves by facilitated diffusion, which I have already talked about. And lastly, this blood, which carries glucose and other products of digestion, they move through the submucosa and the lacteals and everything into the small intestine. This blood is carried via the hepatic portal vein to the liver, where excess glucose is stored as glycogen, which I will cover in subtopic 6.6. If you have learned something, I'm really happy about that. If you haven't, that's okay. I wish you the best with your IB biology exams. And if you need any other videos, just leave a like, subscribe, and share it in the comments. Thank you so much. Bye.